Well, the pleasure is all ours. Thank you for Chris and for Sonia for inviting us to speak at this symposium. We are complete neophytes to ME/CFS, and we've really only very recently started applying our methodology to the ME/CFS space. I'm joined by two of my colleagues here. I've got Dr. Sharoni Das, who's our VP of Bioinformatics, and Christina Taylor, who's our Senior Portfolio Manager. They've both been involved in this study. If you want to know more about the details of them, they're much better to ask than me, quite frankly. So PrecisionLife is a precision medicine analytics business. We are a company. Sorry for that for everyone who doesn't like that. But we have spent most of our time looking at novel ways of analyzing complex chronic diseases. And we come at this from a slightly different perspective than the traditional genetic analysis for a couple of different reasons. Number one, we like multimodal data. We like data that includes both genetic, clinical, transcriptomic, epidemiology, and other types of features as well. And we think that a lot of those triggers are really important to consider inside complex diseases. But the other piece is that we don't accept the clinical label as necessarily meaning that the whole population with a disease has the same cause. So we come at this from wanting to do high resolution patient stratification. In the center here, you see Alzheimer's disease. And what the colors are showing are different subgroups of patients whose cause of the disease is different from each other. And we actually have in Alzheimer's, we've segregated that population into 6 distinct mechanisms causing disease. And we can delineate those by patient stratification biomarkers, genetic in this case. And we also have novel drug targets that can address each of those different patient populations. And that's really as a business where we're coming from. Now, that clearly implies that we're doing things a little bit differently, and we absolutely are. You will see that we're reliant very heavily on the same types of sources of data as everybody else, and we're really using the insights both in terms of novel pharmaceutical development and also in terms of more precision healthcare tools. It's interesting looking at COVID. I wasn't going to tell you all about our COVID work, but it is an interesting case study because it is what got us into this space. We were hunting around for a way to contribute very early in the pandemic, and we wanted to look at the interaction of the host genome. And we did a study on the very first data set out of UK Biobank, and we identified from a relatively small case number a series of really interesting signals. Now, I'm not going to spend a lot of time talking about those, but what I will say, aside from the fact that a lot of these have subsequently been validated by other studies, is that we were finding a lot of the mechanisms, the diverse mechanisms underpinning different symptomology that you would expect. So yeah, viral binding and replication, regulation of inflammatory cytokines, lipid storage, and plasma cell membrane repair, associated perhaps with fusion macrophages. A lot of genes associated with endothelial cell function and microcoagulation, as we saw. A lot with apoptosis and senescence, long-term inflammation, and then others with neurodegenerative disease. And we've done these studies in about 40 different disease areas. So one of the things that we get to do is look at the overlap between different disease states and see what the common pathophysiological drivers are. And that's really where we're starting from. So I said I wasn't going to spend very much time at the beginning, What I do want to do, I'm just going to say this, we do not have time to go through the methodology in a sufficient level of detail to convince computational genomics people in the room, statistical geneticists in the room, but I will explain basically what we're trying to do. So a standard genome-wide association study is really looking for SNPs or mutations that occur more in a patient population than in a control population. And that's great, but it's looking one gene at a time. What happens if multiple genes come together and their combined interactions give you an unpredictable effect on the disease, on the phenotype? Well, that's what we set out to build a system that tests. And so we look for these types of features where you get 6 SNPs all together, and together they contribute to a particular change in metabolism that goes on to cause disease. And a very good example of that is, this is a real example. So these 6 SNP signatures occur in 150 women who have BRCA1 mutations that would normally be thought of as cancer causing for breast and ovarian cancer. But when the women have these particular 6 SNPs, they don't get breast cancer or ovarian cancer. So it's a protective effect. And that gets interesting because that then allows you to start homing in on why that should have that effect. You know, you can start to test a hypothesis for which genes are involved and how you might go about targeting those. So that, as a starting point, is basically where we're coming from. And I'm going to hand over to Shioni to talk about how we use this in the context of ME/CFS.
So we use a hypothesis-free method to generate these SNP combinations. So we call them as disease signatures. And you can imagine for reaching the GWAS significance threshold, the SNPs in a standard genome-wide association study would have to have a large size effect in order to reach that significance. But because we're looking at combinations, we look at significant associations in groups of people. And that is why even if there is diversity in etiologies or even clinical presentations of the disease, we are able to find that signal that is associated with driving the disease in different patient subgroups. So what we do is collate all of the disease signatures that we find are significant in a case population and cluster them based on the patients that are found. So all of the disease signatures would not be found in all of the cases or a majority of the cases, but each of these disease signatures would have a case prevalence. So some of them might be prevalent in 20%, 30%, or 80% of the cases. And we are able to look at that information. So this presents a disease architecture view of the entire case population. What this allows us to do is connect the SNP information. Sorry, forgot to mention that each of the circles that you see are SNPs, and the lines that you see are connecting to 2 circles are showing that they have been found co-associated in one or many of the disease signatures. So because each of these circles are SNPs, and then we have information that the disease signature is found in certain groups of patients, we are able to connect patients through to the genetic signal, which is SNPs, and then we can do all sorts of bioinformatics things where we are mapping them to genes, and then we get information on how multiple genes that we find in our disease network are interacting together that might have an effect in driving the disease or causing different clinical presentations in patient subgroups. Another thing is that because we are able to map to genes, we can bring in information on known drugs that target those genes, which allows us to look at repositioning targets very easily and systematically, and also find out the relevant patient subgroup where we think they might be more effective. So what we do using the disease architecture is try and find the critical SNPs which are critical for driving different biological mechanisms. And these are generally the SNPs or the circles that are found in multiple disease signatures. Those are the ones which are highlighted in green. Here you can see that all of them tend to be the hub nodes which are connecting different disease signatures together. What we then do is score them and prioritize them based on how well they differentiate the cases and controls. This view also allows us to identify communities of SNPs which would have shared biological mechanism, and also allows us to connect them back through to the patients for which we have, if we have clinical or phenotypic data, we're able to connect that information through to these communities of SNPs. What we do on a systematic basis is to look for what is the shared mechanism of genes, which allows us to look at potential mechanism of action hypothesis. Because we have more than one SNP and more than one gene to look at within each of these communities, which represent patient subgroups, it allows us to generate much more insights on the functional and the metabolic context of these patient subgroups, which gives us more insights into what could be a targeted therapy that would be effective in that patient subgroup. Very quickly, for all of the SNPs that we find, especially the ones that are critical in our disease architecture, we map them to genes. We do it as a mapping cascade where we look for is it part of the coding region of the gene, then we map it there. If not, we look at what are the closer genes, and then we assign those SNPs to those genes. If we have information on relevant cells and tissues, we bring that information in as well. But for particularly this study, which is on ME/CFS, we didn't do that because of the uncertainty of the tissues and the cells that would be relevant in different patients, because they would have different clinical presentations and symptoms, etc. We do a lot of bioinformatics analysis where we collate all sorts of information that we can get on the SNPs, targets, drugs that might be relevant in order to interpret what is happening in different patient subgroups. And all of that data is used in a knowledge graph, which allows us to actually look at the potential causability and druggability of these genes, and then develop more targeted approaches in generating evidence for that. Just going back to the disease architectures, because this connects patients' genetic signal, which is SNPs, and then back to genes and drugs, what we are also able to do is correlate any phenotypic clinical data that we have on the patients and see where do they show any enrichment within the communities that we've identified, or are those communities very different or similar to each other. So we can do that based on categorical variables such as like gender or comorbidities, but we're also able to use something more quantitative such as blood data if available. And I'll hand over to Christina to talk about the ME/CFS study.
Yeah, so for the ME/CFS study, we took the available relevant characteristics or clinical data that was available in the UK Biobank with the aim of generating a more clinically relevant patient profile for the different genetic subgroups of the disease that we'd identified. So looking to see if there was a potential viral trigger to ME/CFS development through things like seropositivity for various different viruses, as well as ICD-10 coding for other prior viral or bacterial infection, any comorbid autoimmune disease, and any other environmental factors that might be causing significant stress to the individual before the onset of ME/CFS. And as Shioni mentioned, we did also use some of the available biochemical data in the UK Biobank to look to see if there was any derivations from the average for some of the most common laboratory values that were found in the UK Biobank. Okay, so all of the different studies that we're presenting here today are based out of the UK Biobank dataset. We accept the limitations of the UK Biobank. However, it does still create a very useful way of developing early signals that we're identifying for this ME/CFS population that we're really hoping to validate and expand on with larger patient data sets in the future. So the primary data set that we used for our analysis was derived from the experience of pain questionnaire. So we selected cases of European genetic ancestry only, who had replied yes to the question, ever had CFS or myalgic encephalomyelitis. And we selected a control population also of European genetic ancestry, who had no evidence in any of the hospitalization, primary care, or self-reported data for any evidence of chronic fatigue, post-exertional malaise, post-viral fatigue syndrome, or any other forms of myalgia. And due to the heavy female bias that's seen in the cases with ME/CFS, that control population was also gender matched against the selected case population. Every data set that we generate at PrecisionLife undergoes a standard QC procedure, both for the SNPs and the samples, to remove things like batch effects, filtering for minor allele frequency, and filtering the samples based on factors like relatedness, sex discrepancy, and extreme heterozygosity. And so this formed the primary data set that we will be presenting the majority of the results from. It was made up of just over 2,300 ME/CFS cases and a series of 4,700 gender and ethnicity matched controls. And just to point out, there is a secondary way of classifying ME/CFS within the UK Biobank. That is through the verbal interview that was conducted upon recruitment to the assessment centre for the UK Biobank. And so we were able to use this as a secondary data set with which to compare the outputs from that primary pain questionnaire cohort. And so these were included those cases that had answered yes to having chronic fatigue syndrome, but were not included in the pain questionnaire case cohort so that they could form an independent group with which to compare the results against. And these were compared against this, a very similar set of controls with the exact same criteria and underwent identical dataset QC measures. Shane?
So the first thing that we did was look at what we would get from a standard genome-wide association study. Here you can see that we do not see any SNPs that reach a significant threshold. And what we also saw, which we have not shown here before, this is from the pain questionnaire dataset that Christina just mentioned. We also had the secondary dataset, which was the verbal interview dataset, where we did a similar analysis of GWAS. One of the interesting things that we noticed was, even though in both of the data sets there was no GWAS significant results, the SNPs with the lowest p-values, which they were both pointing to different loci in the two data sets. So if it was blinded, it was very difficult to know that they came from the same clinical diagnosis. What we did find using our combinatorial analysis was 84 significantly validated disease signatures in different groups of patients within the cohort, within the pain questionnaire cohort. One of the main things I would like to highlight, the histogram shows that out of those 84, all of them had at least 3 SNPs in combination. So each of those 84 disease signatures had at least 3 SNPs in combination. So we are able to map at least 3 gene regions for each of those disease signatures. And that is adds a lot more power to the bioinformatics analysis, because instead of one particular SNP that we go ahead and look at what might be influencing that, we have 2 or 3 gene loci that we can look at what may be connecting them and what influences it may have on the patient subgroup where we find that significant. We looked at, so these 84 disease signatures were comprised of 199 SNPs. Some of them were found in multiple disease signatures, and that resulted in 25 critical SNPs that we have prioritized for analysis, and they mapped to 14 genes. When we looked at what these SNPs were, we can see that 95% of them came from the non-coding. They were all non-coding, and only 9 of them were coding variants. None of the disease signatures that we found, out of 84, none of them were found to have any SNPs in linkage disequilibrium or in LD. Another thing I would like to point out is the odds ratios of the SNP combinations that we identified, which is 3.7, which is higher than a normal GWAS analysis, because here we are looking at a combination of SNPs, which is significant in much lower, relatively lower number of cases than what would achieve dual significance. This shows the disease architecture of the ME/CFS pain questionnaire population, which comprised of the 84 disease signatures. Here, each circle again shows an individual SNP, and the ones that are highlighted in green are the 25 critical ones that we have mapped to genes and prioritized for analysis, which we'll be talking about. One of the things that we've looked at is these are all segregated patient populations, so segregated groups that you can see in the colors. We looked at the patient overlap between these patient subgroups and saw that they were quite distinct, and less than 20% of the patients were overlapping with any of the other patient subgroups. So that was quite interesting to note that these groups could potentially be, could have different clinical presentations or phenotypes that we were going to investigate.
Okay, so one of the main ways that we validated the results of those 199 SNPs from the pain questionnaire cohort was to compare it against a series of independent replication cohorts made up of both the verbal interview chronic fatigue syndrome population, but also 2 other fatigue-associated disorders within the UK Biobank. So the G93 post-viral fatigue syndrome ICD-10 code, and also the ICD-10 code for fibromyalgia. And as Shoni alluded to a couple of slides ago, even though the GWAS Manhattan plots for all those 4 cohorts looked entirely different, we do see some replication in significant SNPs that are coming out of those other replication cohorts when they're compared against the original pain questionnaire, with 5 of them being found in the verbal interview chronic fatigue syndrome data set. So for us, that is great validation for the significance and the relevance of those critical SNPs that we identified in that primary analysis. So the next series of slides just takes you through from start to finish that pain questionnaire analysis. So as Shoni said, SNP genotype data from that 2,300 ME/CFS cases and the 4,700 controls was inputted into the PrecisionLife platform that generated the disease architecture on the slide. That generated 25 critical SNPs, which using our standard SNP to gene mapping process could be mapped to 14 protein coding genes that I then start to analyze and think about what mechanism of action hypothesis that could be, or why variants in those genes could be impacting the development of ME/CFS. Those genes do undergo a series of validation. All of the results are evaluated for things like LD effects, as well as the odds ratios and the p-values of the SNP combinations identified are very thoroughly evaluated. And we've also, as we said, compared the results against a series of other disjoint cohorts, as well as performing a smaller cross-validation study where we take subsets of that original pain questionnaire cohort and evaluate the significance of those same SNPs in those smaller subgroups. So onto the biological annotation of what those genes actually are. We admit that is a subjective art form. However, as Shoni has demonstrated, we bring in a very big variety of different data sources with which to annotate and evaluate those genes, and a constant set of heuristics is used to evaluate each to try and remove some of that element of subjectivity. And what we've been able to do is categorize broadly the mechanism of action hypothesis for these 14 genes, which can broadly be split into 4 main pathways. So those with perhaps dysregulated host response to viral or bacterial infection, those with metabolic dysfunction, as well as sleep disturbance and autoimmune development pathways. And given that we don't have time to go through all of those 14 genes in detail here, although they are described in more detail in the paper that we've written, I'm going to take you through 3 examples of some of the genes that we identified here. So the first one, patient subgroup we're looking at is with the patient subgroup ID 9, and that actually made up 648 cases from the 2,300 that we originally inputted. So just under 30% of that ME/CFS population. And as you can see, it's got an incredibly low p-value and a high odds ratio. And the core SNP that is found in that group or combination of SNPs maps to the gene ACAP1. And we'd categorize that as being in the metabolic dysfunction category from the previous slide. And that is because ACAP1 regulates mitochondrial respiration via AMPK, and actually phosphorylated ACAP1 is necessary for AMPK-induced increase in respiration post-exercise. So we can start hypothesizing that variants in this gene might be involved in that post-exercise recovery or impaired recovery that is seen in ME/CFS patients. And actually there is a paper out there that's looking at AMPK that has found that activation of that protein is impaired in cultured skeletal muscle cells that are from the ME/CFS patients. And finally, there's mouse knockout data as well that shows that reduction in ACAP1 actually reduces the respiratory capacity and skeletal muscle capillary density in those mice. So in that sense, we're building up this picture and this mechanism of action hypothesis that patients that are found within this particular subgroup are likely to demonstrate some of the dysregulation in mitochondrial respiration that is sometimes seen. The second subgroup that we're going to focus in on is subgroup 15, that has the MAPS gene SLC6A11, and that encodes GAP3, which is a GABA reuptake transporter. And various different animal models of mice and rats found that altered GAP3 levels are associated with things like cognitive impairment, vulnerability to stress, and sleep disturbance, all of which are experienced or can be experienced by ME/CFS patients. And this is where we track back to that clinical phenotype of this particular genetic subgroup. We found that cases in this subgroup had a significant association with increased phenylalanine levels from the metabolomic data in the UK Biobank. And what is interesting is that phenylalanine is a precursor to the monoamine neurotransmitters like dopamine, epinephrine, and serotonin. So it seems that patients in this subgroup may be more likely to experience some of the neurocognitive effects of ME/CFS. And actually, when we evaluated the verbal interview cohort, the independent analysis, 2 other SNPs in the same gene were also highly significant, which is telling us perhaps it's more of a gene dysfunction rather than an individual SNP effect on ME/CFS development. And the final one, very quickly, is from subgroup 2, and this is a gene that we put under the mechanism of action hypothesis for sleep disturbance. And that's because the MAP gene is the clock gene, which is the key regulator of circadian rhythm. And that's both been implicated in the symptoms experienced by ME/CFS patients, but also the biological pathways like the disrupted mitochondrial respiration that has been associated with ME/CFS. And other analyses have found other genes in that same pathway are also elevated in a transcriptomic analysis of ME/CFS patients. And very finally, patients within the subgroup were also significantly more likely to have an additional fibromyalgia diagnosis. And there has been a separate study that's found that when they look at patients with ME/CFS, those with both ME/CFS and fibromyalgia alone, it is that group in the middle with both ME/CFS and fibromyalgia that are more likely to report insomnia and sleep disturbance. So this might provide some hypotheses as to how genetic variants could be impacting that. And this is just a reiteration of some of the replication of the critical SNPs and the genes that we identified in those separate disjoint cohorts. The final 2 slides. It's more to say there's a lot of detail here that we've both gone into and haven't. So the more detailed set of results and all of the different validation techniques that we used to analyze this population can be found on our preprint on medRxiv. And as has been discussed here, ME and CFS and long COVID obviously share very similar symptoms, and that might point to common genetics and underlying biological pathways. So we've actually already found 3 of these genes from the ME/CFS study in a preliminary analysis of a long COVID patient population data set.
Thank you. So one of the things that we can usefully do with this, because we've got data at a similar level of resolution across 40 different disease areas, going back to the repurposing opportunity, we can look for influences of specific pieces of target biology in clinically relevant subgroups across those diseases and ask the question, are there existing drugs that are known to modulate those targets in the right direction? We can do that systematically now. I'm not going to go into the details of it, but we've got all of the biopharma company pipelines of development candidates and on-market drugs, and also some withdrawn for efficacy that we can automatically analyze these. So I think we've got to ask, where does this take us? I think there's a couple of interesting things for this afternoon's discussion. We'd love to validate some of this with real world evidence. You've got a fabulous resource in Scotland, the data lock. There's opportunity to look at people who serendipitously have a diagnosis that's led to prescription of a particular medicine. And we can look at the downstream effects of that in terms of prevalence of ME/CFS and recovery times, for example. We can look at cross-disease processes. We can look at trying to identify and home in on disease-identified tissues, which would get us into being able to do genetic knock-ins, knockouts. And also, ultimately, we want to get to a point of having metabolomic biomarkers. So I think there's a lot of interesting places that this research could take us. As I said, we're not experts in this. We rely hugely on the community to guide us in this. I have to pay huge tribute to Chris. He has been a fantastic help in steering us in the right direction and in looking at some of the analysis that we've done, and to Sonia also for encouragement to work inside ME/CFS. Obviously, the PrecisionLife team, UK Biobank, we couldn't do this stuff without, and finally, all of the patients without whom none of this research would remotely be possible. So thank you very much for your time.
Excellent. More chairs hands, everyone. So thank you very much. Okay, so in the room and online.